recording now. A couple of announcements for this week. So in lab this week, part of you are going to start the light reactions. Group B is to do the light reaction exercise in lab, plus you're going to be taking data on your hormone lab exercises. That's going to be a busy lab, both this week and next week. And so Wednesday's lecture, I will be, part of the lecture period, I will give the lab lecture. And so I will give the lab lecture, and then when you walk in, Dr. Chu or Laren will not be really lecturing about it. You'll just get going on the actual experiment, all right? So that should shave off some time to allow more of the full 3 hours for everything that needs to get done. So Wednesday's lecture, I will be doing a lab lecture, and then after that, I'll get back into content for the lecture. So lab lecture and lecture on Wednesday. I'm behind when I look at the syllabus a little bit, and I think I'm going to push the exam because of that. The next lecture exam is already on the syllabus. It's on the syllabus, it's stated that it's the week of next week, but I'm falling behind. And so I think I'm going to push the lecture exam to the following week, which would be the week of October 5th, sometime in there. So that's the tentative plan. Yes, and that's the other thing too, is I have to get the previous exam back to you. We have to go through that before you take the next exam, because I want you to learn from your mistakes. It's one of the best ways to learn. And so I'm still working on the exam. It is slow going with kind of need chunks of time in order to really get into it and get into the swing of it. And it's been really challenging to find chunks of time. So I'm picking away at it, but it's slow. So exam tentatively will be sometime during October 5th, assuming I don't get even further behind. Okay, so let's plan on that and we'll go from there. Okay, I'm going to share my screen and get into off. See if I can do this. Someone has their mic going. It's loud. Share screen again. Okay, so hopefully you're just seeing one big slide. Thank you. This is where we left off. So when you go to buy a bottle of fertilizer at the local store, it talks about nitrogen, phosphorus, and potassium levels typically, NPK. So nitrogen is needed in high quantities because it's nitrate. Remember that there will not be a reservoir of nitrate in the soil system because the soil particles are negatively charged. So nitrate will not bind to the soil. It stays in solution, and which is on one hand good because then the plant can take it up. On the other hand, not so good if you misjudge and over fertilize. You can cause a lot of pollution effects because of that. But anyways, so symptoms: slow stunted growth if you don't apply enough nitrogen, general chlorosis is very common. On the other hand, it's a very mobile element. Once it's taken up by the plant, if young leaves really need nitrogen, the older leaves can sacrifice and send some of that nitrogen they have in their tissues to the younger, more active growing parts. And so the deficiency symptom will show up where the old leaves start going chlorotic first. The young leaves stay green. So look at that. If you have houseplants, really start trying to key into what parts of the plant body are showing symptoms. And that can go a long way to helping you diagnose if it's a mobile or immobile nutrient when that nutrient is in the plant body. This is not something I'm going to ask details on in the next exam. However, I just want you to appreciate that, you know, nitrate is the preferred source, but there are other forms of nitrogen that can be taken up by different plants, and so they have different ways of dealing with it. And so they've evolved mechanisms to be able to take in nitrate, certainly, but also ammonia, ammonium, And sometimes that has to have an expenditure of ATP to get that to happen. But you know, it's such an important molecule for the plant body, for the structure of the plant body, that it's no surprise really that they have evolved many mechanisms to deal with this. A little bit about phosphorus. That's the second ingredient, so to speak, on bottles of fertilizer. And the H2PO4 minus form is the one that is most easily absorbed by the plant. Phosphorus is tricky because phosphorus in the soil often is complexed with something else and precipitated out. So even though you on the face of it, if they take a soil test and don't do it the right way, they might say, oh yeah, you have a ton of phosphorus, no problem. But you have to do specific ways of testing to show what's available and what's precipitated out and unavailable. Rise above pH 7.2 and the phosphorus tends to complex out and become unavailable. And so in general, it's a safe bet to say a general rule of thumb is that the concentration of soluble phosphorus, which means it's dissolved in the water, the concentration of soluble phosphorus in most soil systems is low, whether it be your garden in your backyard or an ag field. This is just something that's pretty well known, and it's unfortunately pretty easy to get precipitated out under super, you know, above 7, but even super acidic conditions too. So it's a tricky one to work with. If you get it in your if your soil pH is 5.5 to 6.5, it's good. It's pretty good to get phosphorus that way, but below that and above it, you got some problems. So for example, if you have acidic soil or above pH 7, 7 and above, it tends to, depending on the soil composition, aluminum loves to complex with phosphorus, and so does iron. And then you'll, if you push above 7 into like pH 8 and above, then you have magnesium and calcium interacting with it under those conditions, and it will precipitate out. So what should you look for? It's weird because you'll look at a plant and you go, wow, those leaves look really green, and you might think, oh, it's doing great, it's super healthy, but be careful. It could actually mean that you have a phosphorus deficiency. I've been fooled when I look at a plant that I'm growing for a study, and it's like, oh, it looks so green, this is great. And then we get the analysis back on the tissue, and it's not so great. Necrotic spots can pop up. So kind of a mottled necrosis can start happening. And they also will exhibit that purpley red pigment on the underside of the leaves or on the petiole, which is just the little stem of the leaf blade. And so that is something that you can look for, and it's not all plants will do it, but if you do see that, then that should ring a bell. Uh-oh, we either have phosphorus or nitrogen deficiency going on. Once it's in the plant, it is mobile. So younger leaves, if they have a high demand for it, they can get it redistributed to the younger leaves. So that means the old leaves are starting to show symptoms first. Now, because phosphorus is such a pain to get into solution, plants have evolved mechanisms to try to deal with this, but not all plants can do this, and it's with limited success. So this image here is getting at that. And again, you know, I'm not going to get into the complex chemistry of it, but basically what they're showing, for example, in the figure A here, is that the plant will be carrying out respiration just like we do, and you know, we give off CO2, so it'll plant. So the CO2 will diffuse out. When CO2 interacts with water, it forms the bicarbonate ion, and then it can interact. That actually says calcium phosphate, I think, on the soil particle. I don't know if you can see that from where you're sitting with Ca, calcium. And they're just saying that under these conditions, the bicarbonate can react with the calcium phosphate, release the phosphate so that the plant can take it up. You do see energy expenditure to get this to happen. ATP is being expended in order to get this symport process of pulling in the phosphate as the protons are diffusing across. So there's an expenditure in all of these, you're seeing that. Citrate is another thing that they can exude out, excrete out, and get another system in place where it can release the insoluble phosphate. And then an enzyme. Some, not all plants by far, but there are some plants that can secrete a special enzyme called acid phosphatase, and that will also solubilize the phosphate and allow it to be taken up. So there are mechanisms in place, but unfortunately not all plants can do this, and especially some of the plants that we have developed for crop production, they don't necessarily have the ability to do this. And so they really have to rely on the mycorrhizal fungi. So if you have a field soil system that's deplete, there's a depletion in phosphorus, spiking in mycorrhizal fungi will usually greatly help. And in fact, when I was doing work with ectomycorrhizal fungi with red pine seedlings, my one grad student, that was her project, she was looking at the presence, absence of ectomycorrhizal fungi, and then she was stressing the plant in different ways to see the plant response with or without the partner fungus. And we quickly, well, not so quickly, it took a while because these fungi grow slowly, but we realized after her first set of experiments, she couldn't get the mycorrhizal symbiosis to establish. And she dug up all the roots and there's no ectomycorrhizal fungi there. And then we realized we were being too nice to the plant. We were giving it all of its, all the phosphorus it wanted, all the nitrogen we wanted. So when we provided a sweet setting for the plant pretty much said, I don't need a partner. I got all of it right now. And it was very easy for the plant to get access to it. So then we learned, okay, got to be a little stingy and mean to the plant and withhold the phosphorus. And as soon as we withheld phosphorus to low levels, man, we got some really good mycorrhizal symbiosis going on. So understanding, when you look in certain fields that are really amended well, you might not see a lot of mycorrhizal fungi presence. You'll see them there, but not to the same level as a soil that's been deplete in certain key nutrients.
So that area with phosphorus after that
So then the question was, what happens if you take that soil and there is low phosphorus, so the mycorrhizal relationship establishes, then what happens if you spike in a bunch of phosphorus to make the plant happy? They tend to give up the relationship. The plant will say, I don't need you anymore, essentially. And and they've done this where they can track the presence, absence of mycorrhizal fungi and percent presence and whether or not they've truly established with the root. And they break the relationship essentially. So yeah, very interesting dynamic. So phosphorus is a big one to manipulate if you're looking at working with mycorrhizal fungi and getting them to form that relationship. Potassium is the third big player on bottles of fertilizer, so I wanted to mention that one a little bit. And it's very easy to amend into the soil system. It's brought up as the ion and into the body, and potash is commonly applied into the fields. You need a lot of potassium. We're going to find out that potassium is very important for the plant, but it's super soluble in the soil system, in the soil water system, so it's usually not a huge problem, except if you have big soil particles like sandy soil, then the potassium, because it's so soluble, it tends to leach out under sandy soil conditions. But once it's in the plant, it's highly mobile, very highly mobile. So again, the older leaves will sacrifice or give up some of the potassium to younger parts of the plant body if needed. If you're out in a field, for example, and you're looking for potassium deficiency on the leaves, you would be looking, mottled chlorosis is a really strong indicator of a potassium deficiency, and then that can turn into mottled necrosis over time. And then look at the tips of the leaves and the margins of the leaves for necrotic or chlorotic tissue. And that's another giveaway that you're low in potassium. But beware, that can also mean something else. So often plants that are in too salty, saline soils, if there's too much salt in the soil system, they will take up some of those ions, but it's like, oh, this is too much salt. And so they'll actually preferentially pack it into cells that are on the tips of the leaves or the margins of the leaves, because those are kind of like the first that they sacrifice for storage. And so be careful about that. My one grad student, I had a potted plant in my office, Scott.
That's way quicker than the natives would be.
Daniel, could you mute yourself, Daniel?
Oh, sorry.
That's okay. So the pot had all these white crusty things on the surface, and on the face of it, I just kind of glanced at it. I thought it might be fungal hyphae. I didn't look that closely. And he grabbed the pot and he's like, oh, what's this? And he licked his finger, touched the white stuff, and then licked his finger again. I'm like, Scott, what are you doing? He's like, oh no, that's salt. And I was yelling at him like, you can't do that. And he's like, oh, I do it all the time. That's just salt. So salty soils, the salt and your potted plants can actually accumulate and float up to the surface, so to speak, and crystallize. And then it's like, oh boy, this poor plant. And sure enough, when I looked more carefully, the leaves, margins, and tips were just brown and dead. And so I unpotted the plant and essentially flushed the soil with distilled water thoroughly and then repotted. The other thing too, in ag fields, you'll see these weird, the plants will actually like topple over. So the shoot will just kind of, the stem weakens and then it just topples over, and that's called lodging. They actually had to come up with a word instead of it tipped over. So they call it lodging. And so what's happening is the stem tissue gets weak enough. And then of course, that's a huge problem if you're a farmer, because the machines they use to harvest don't deal well with lodged plants. And so it's a huge problem for growers. Plus the plant is suffering and you won't get as much cereal seeds as what you should. So big problem with potassium deficiencies. But usually if you know what you're doing, you can very easily keep potassium levels at a good dose. Okay, so there's several other slides here walking through some of the other macronutrients. I'm not going to go through them in any detail in lecture. I think now you understand kind of what my bullet points are pointing to and what I mean by that. But there's a few others I just want to mention. Let me go down to, I wanted to mention magnesium. I have to stop share, don't I? Oh, it's working? Oh, amazing. All right, so magnesium, I wanted to mention this one in particular because it's really needed in high amounts. And sadly, so much of our ag soil is fairly deficient in magnesium. I did enough reading a few years back that I'm not a huge fan of supplements for my own diet, but magnesium is one I regularly take because it's needed for so many of our physiological reactions, including our ability to make ATP. And when I read about how poor our ag soils are doing in terms of providing enough magnesium, I'm not trying to tell you what to do. I'm just saying that that's something that I have taken pretty seriously. And it's super important for the plant too. I mean, so much of what they do, and then the chlorophyll molecule is also, there's a magnesium chelated right in the chlorophyll molecule. And obviously if they don't have enough magnesium, then they can't make chlorophyll, then they can't do the light reactions of photosynthesis. So it's super important. It impacts ribosome function, the ATP production, and so it's involved in our bodies as well as in the plant body in so many different key important reactions. Obviously we don't have chlorophyll, but for them it's a big deal. And then we'll talk about rubisco and phospholinopyruvate, hugely important enzymes. Rubisco is the most abundant protein on the face of the earth because all photosynthetic plants and other photosynthesizers have that. Intraveinal chlorosis is classic. So if you see the veins popping green and all the tissue around the veins is pretty yellow, that is one, right away you should start be thinking about magnesium issues. It is mobile in the plant body. So again, the older parts of the plant will display deficiency symptoms first. The younger parts will be okay. And like I said, so this is chlorophyll, the structure of chlorophyll, and we'll learn about this again, I'll point it out again, but basically this tail region of the chlorophyll molecule, that's what embeds into the thylakoid membrane inside the chloroplast. So that's embedded across the membrane, and then this head region of the chlorophyll molecule sticks up off the thylakoid. But that, you can see magnesium right there and is part of the structure. And if you do not have this magnesium chelated in the head region of the chlorophyll, it's just useless. It won't work for the light reactions. So really important. By the way, if some of you have taken animal or human fizz, that structure may have looked somewhat familiar because chlorophyll and hemoglobin have a very, very similar makeup. Iron in our hemoglobin is what's chelated in the middle, but with plants, it's magnesium for the chlorophyll. So really interesting similarity there to think about. But, okay. The micronutrients, make sure in the book they talk about the micronutrients as well. I think each nutrient gets like a little paragraph in the textbook, so make sure that you're reading through that. And those tables, remember that table 4.1, I think, that groups them out by their activities and what they do for the plant. So make sure that you're working through that information as well. There was one other, what was it? Magnesium, oh, iron. Wanted to mention iron. Is it still working when I pop up iron? Okay. Iron, so I have written down here Fe2 plus and Fe3 plus. So the Fe2 plus is ferrous, the ferrous form of iron. And the Fe3 plus is ferric, F-E-R-R-I-C, ferric. And it's important that you know the difference in terms of the Fe3 plus, the ferric form, is really insoluble in water. It is, if you work with it in the lab, I often have to add iron to recipes when I'm doing tissue culture or something where I'm growing up plant cells. And oh, is it a pain to get into water solution? You have to heat it up and it's just a pain. We often have to add in chelating agents in order to deal with it. Of all the micronutrients, probably iron is the one that is needed in largest amounts by the plant. And they're really important in a bunch of different redox reactions that happen in the plant as well as in our body. And also the production, the synthesis of chlorophyll pigment, you need iron for part of the process of the physiological process of building that. It's also showing intervenal. So magnesium and iron can show similar intervenal chlorosis, so you have to be a little careful. However, iron is immobile, so that can help you differentiate because magnesium is highly mobile. And so that means the older parts of the plant bodies will display symptoms with magnesium. With iron, it's the opposite. Young parts of the plant will show intervenal. So that's one way you can tease apart the difference if you're thinking about magnesium versus iron deficiency. Too much of iron can be bad, just like with us. Adult males in particular, humans, you have to be careful not to get too much iron. It can actually be not so good for you. It's usually not a problem with females because of the menstrual cycle. You lose a lot of iron. But toxicity for the plant can be strong in acidic soils. So below the 5.5, the plant can take up too much iron, and that can be a problem. But deficiency is common. That's way more common than too much. And so again, the ferric form, Fe3 plus form, very quickly precipitates out in anything, you know, outside that 5.5, 6.5 range. You go above it and you can get that insoluble oxides forming. So plants have evolved mechanisms because they need iron in such high levels that it depends on the plant species that they studied, but some plants have a way to chelate the iron, so they produce a chemical that very readily latches onto the iron and kind of sequesters it away from other organisms, and then they're able to absorb the iron into the root. And so if you're in chemistry classes, they may refer to it as a ligand or a ligand, depending on who's talking. I tend to say chelating agent. So it's the same thing, but basically in the lab, you can do it by spiking in EDTA. So when I'm working with iron in the lab, I'll often have to add in EDTA, which is a chelating agent that will help the iron go into solution better. Plants will produce caffeic acid, and not all plants, but many can. And caffeic acid is the chelating agent, latches onto iron and helps the plant kind of sequester it away from other organisms, and then they can take it up. So here you're just looking at the DTPA actually in this picture. I'm just trying to show you how it's chelating and bonding with the iron ion. And so if you're curious, that's what I mean by chelating. You have that very particular bonding pattern. So some plants can do this with caffeic acid. So there's a structure of caffeic acid. And I'm not going to get into the details of it. If you can understand that caffeic acid is used for iron scavenging and to chelate that, that's good. But plants definitely, some plants have evolved this mechanism so that they can get access. And usually what happens is the caffeic acid can get recycled. So when the caffeic acid binds to the iron and brings it to the root surface, usually the plant will just cleave off the iron and the caffeic acid will stay outside the root to continue to keep doing that, go bind more iron. And so it's a process that's been looked at pretty heavily. Another way to deal with getting access to enough iron, and these are studies that have been done in the mostly in lab settings. If you put a plant deliberately under iron stress, some species, not all, but some plant species produce what they now call phytosiderophores. And anytime you see PHYTO, phyto, in a word, that means plant or plant-like. So siderophores were first discovered in bacteria. In fact, pathogens that attack you, microbes that make you sick, usually have a high demand for iron. And that's one of their strategies. They will actually send out siderophores into your body and latch onto iron that's in your body so they can have it for themselves. And so you often get depleted in iron when you get sick by some of these pathogens. And that's why our response to that is to spike a fever when we get, that's part of our immune response when we're getting attacked. But one of the good things about a fever is not only is it helping you just with the immune system gearing up, but specifically, apparently, what I read is that the fever kicks in a pathway, stimulates a pathway in your body that produces special proteins that you make that will latch onto the iron and keep it away from the microbes. So it's a way for you to, essentially, you're chelating the iron in a way. You're binding those proteins you're making are binding the passage of the iron so the pathogen cannot get access to it. So when they discovered this, not too long ago, really, like when my boys were little and they'd pop a fever, our doctor would actually say, just let the fever go, let it, monitor it, but let it go because it's actually a beneficial thing. And so, and I would agree with that. In fact, my grandfather, he was 100% Finnish and he wasn't a doctor, but he seemed to just through life experience know whenever he start feeling icky and like, oh boy, I'm coming down with something, he'd fire up the sauna and take a blistering hot sauna. And he swore that that would help, but that's essentially what he was doing, right? He was elevating his temperature like a fever. Seemed to work for him. He lived a long life, so he didn't get sick very much. So these plants can do it now too. They said, they knew bacteria could do it, and they're like, oh, plants are doing this too. So they named them phytosiderophores, which are really good at latching onto the Fe3+, which is the most insoluble or difficult one to work with. And then it seems like the whole thing comes into the root, though. They don't cleave it off. The whole phytosiderophore, at least with some of these plants they've been looking at, will get reabsorbed by the root. So it's a little inefficient in my opinion, but it works well enough, I guess. So here they're just trying to show this idea of whoever the artist was. I don't think the protein's actually called yellow stripe 1. Do you see that? So apparently they forgot to label what this protein is. Yellow stripe, that's just referring to the artist's rendition. But anyways, this is just trying to show the idea of the chelating of the iron and the conversion of the iron to the ferrous form, and then it can be brought in. And then here's your siderophore complex, where you can have this absorption going on. The whole complex gets absorbed through a special transporter. All right, so iron is super important. It is a micronutrient, but I've been privy to some conversations with plant physiologists where some are saying it should be a macronutrient. We should shift that, but they haven't done it. I don't know if they ever will. All right, so then make sure that you're reading in the textbook on the other, oh, and then beneficials. Pardon? Yeah, I know. It's like, that's not. No way.
Iron.
It can't be.
Siderophore, transporter, yellow stripe 1.
I think that's a typo. I think that's the artist. They didn't fix it. It can't be yellow stripe one. Okay, now I gotta find that out. That's gonna bug me. I'll do it after I grade the papers, but that's gonna bug me. All right, so beneficial. You'll hear some plant physiologists argue about beneficial elements. And so there's this nebulous group of elements that we know not all plants need, but some do, and need some of these minerals. So sodium, actually, most plants really don't need a lot of sodium, and sodium can quickly become toxic to them. But there are some plants that have adapted to high saline soils and actually now do require it. Silicon, if their body has a lot of silicon in them, then that's something. So again, it depends where the plant is adapted to, where it evolved to live, and whether or not these elements are impacting their, speciation and evolution of their ways that they deal with these different minerals. So be aware that not all plants, there are some special cases here. And then I put this in from a different source, looking at deficiencies, if you're interested in some other sources that are looking at deficiency symptoms. And then I put in a couple slides looking at what happens if you get too much, because too much of anything can be bad for you. Same thing in the plant world as well. So just more for, your own knowledge, you can look at some of these ideas of what happens if you overdo it. Okay, and all right, so mineral nutrition, hugely important for the plant. And so, it plays different roles in the plant, anywhere from, building the plant body to redox reactions and their ability to get their ATP needs met. So really, really important. Okay, we're going to stick with minerals in a way, because the next thing I want to talk about is biological nitrogen fixation. So is it showing up as one big slide again? Oh, okay, I don't know why this is so successful, but anyways, so biological nitrogen fixation. So we're going to stick with nitrogen, but now we're going to talk about how plants have partnered up with certain bacteria to be able to get access to nitrogen that is super abundant, right? We have what, 80% of dinitrogen in our atmosphere, and most plants, well, plants at all, there's no plant that can touch it, only microbes. And when I say microbes, I mean specifically bacteria, and not all bacteria, only very special bacteria. Fungi cannot do this. Mycorrhizal fungi cannot do this. Is everyone listening to that? Mycorrhizal fungi cannot do this. Think that might show up on the exam? It's one of my pet peeves. I'll just let you know. Only bacteria so far. If you can discover a mycorrhizal fungi that can do this, you'll probably get the Nobel Prize. All right. So as far as we know, and they've looked pretty exhaustively, only certain types of bacteria. So they're prokaryotic organisms. They have never found a eukaryotic organism that can fix nitrogen.
So
you can engineer it, maybe, and they're working on it. They're working on it. It'll be interesting. I bet in your lifetime you'll see it, but naturally. So prokaryotic only certain types of bacteria, really special ones. It's really a cool thing. So what are we talking about? So the nitrogen in our atmosphere is N2. That means it's dinitrogen, and it has a triple bond in between those two nitrogen molecules. So dinitrogen has a very special triple bond. Well, triple bonds, if you remember chemistry, pretty hard to break that bond. It really needs some special effort. So this is the enzyme that can do it. It's called dinitrogenase. A lot of people just shorten it down and just say nitrogenase. Either one is fine in my book. And so dinitrogenase is the enzyme, and only prokaryotic cells seem to have this, and only some. So what does it do? It breaks the triple bond between the two nitrogen atoms, and the immediate product is ammonia. Well, ammonia is toxic in high doses to all of us. That's what's in smelling salts. When you pass out and faint, they'll crack open ammonia in the smelling salt, and your body immediately jerks into consciousness, because you're hardwired to run from ammonia because it's so deadly. So it's kind of weird, but what they do is they'll take the dinitrogen, make ammonia, and then they quickly convert the ammonia to something else. And so it doesn't persist in the body. They're not going to die from it. This is actually a really good thing that they're doing. So what are the bacteria that do this? There's a group name that we give them. We call them rhizobia. All right, that's just kind of a general term. That's not a genus name or a phylum name. It's just like a group name. We say, oh, you know, that's a rhizobiobacteria. It can do nitrogen fixation. Rhizo referring to root. So they, you know, we're thinking about the, and I'm going to zoom in on the bacteria that hang out with plants, obviously, because this is a plant phys class. But there are bacteria out there in the real world that won't hang out with plants necessarily, and they can still fix nitrogen. So you don't have to partner up with a plant to do this, but many do. And then there's some that will do it on their own, but then when they hook up with a plant, they'll do it with them too. So there's some range in how this is done. But the rhizobiobacteria are the ones I'm going to be focusing on and telling that story, because they're the ones that definitely do connect with the plant root and start this relationship and process. So these are some of the genus names that you would see in the published literature: Rhizobium, Bradyrhizobium, Azorhizobium. There's many others. If you do a Google search on nitrogen fixation, there's literally thousands of papers that have been published on this topic. Really active area of research, especially when you look at crop production, because they want to, you know, a lot of scientists are like, we should make more plants have this ability so that we can, you know, cut back on nitrogen fertilization and improve crop yield and so on and so on. So it's a very active area of research to this day. I don't see it ever dying off anytime soon. So what would you look at? What would you see visually? This is some pictures showing the idea that if you carefully dig up some plant roots and you see these little bumps, and you know, when I was little and I dug up some of these in my mom's garden, I thought, oh, this poor plant, it's got a tumor. But these are the nodules that you would see, that it's plant tissue. So the plant, and I'll talk about this, but basically the plant overproduces certain parenchyma cells in this area of the root system, and it creates a house, a home for the bacteria to hang out in. So it's a way for the bacteria to be housed safely in the root tissue. And then there's connections, you know, vascular connections that feed into the nodule, kind of like tumor, you know, how blood vessels feed into a tumor. There's some analogy there, but this is all good. This is a really good thing. And they look a little different depending on the plant species. You can actually see on the right picture that the nodules look a little pink. I'll talk about that. There's a really, really interesting partnership that gets developed in order to make a very particular protein that, again, is very similar to hemoglobin. And they actually named it leg hemoglobin because legumes, the type of plants that are called legumes, are able to form this relationship with bacteria and have nitrogen fixation. And it's really a fascinating comparison of how it looks and how it functions, similar to our hemoglobin. But anyway, so here we're seeing another close-up on a bean, which is a legume, and you can kind of see the pinkish reddish hue to it. Again, it's analogous to the hemoglobin idea. Here's some others on, again, showing the kind of the reddish hue. So it just depends on the plant, how big the nodules are, what color, what shape. There's some diversity there. So, and then I just wanted to, you know, there are microbes. This is just a free living cyanobacteria. And so they're pretty darn special because not only, so cyanobacteria, they can photosynthesize, but they can also fix nitrogen. So they're a pretty powerful little microbe. And so they can do that free living. You can see cyanobacteria in ag soils, and people are like spiking them in to see if that would help with nitrogen levels, but I don't know much about that research. That's not my field. But so there are free living microbes and some that can photosynthesize that are able to fix nitrogen. All right, so you don't have to be with a plant necessarily. Bradyrhizobium is with the plant roots, and you can see under the microscope, they don't look like much. They're just these rod-shaped microbes, nothing special to look at, but boy, they're doing some pretty spectacular things in their bodies, in their cells. All right, so this table is from your book, looking at the idea of if it's symbiotic is the top portion of this table. So the legumes on the top row, and you can see a bunch, and this is not a complete list, but you can see a bunch of them there. Frankia, really interesting group. Frankia bacteria are very similar to the Streptomyces bacteria I work with, and they can do nitrogen fixation like so alder trees around here. If you dig up their roots, you would see nodules as well on there. So So there are different microbes with different plant species. And then here you see free living, the ones that just can do this all on their own, and they don't need to be with a plant. All right, so there's some diversity. Okay, and then here that we're kind of zooming in between the plant host and then which bacteria are associated with that particular host. And so now we got to think about this idea of when you have this partnership between the plant and the bacteria that can do nitrogen fixation, it's usually very host specific. Now, when we looked at the mycorrhizal fungi, I didn't really say too much about host specificity because mycorrhizal fungi are pretty easygoing. They'll form relationships with a lot of different plants. The bacteria, on the other hand, that do nitrogen fixation, it's usually a very tight association, and that bacteria really only hangs out with, you know, a very select few, and sometimes just one that we're aware of type of plant. So that means there has to be some very careful communication going on between the plant and the bacteria to make sure that, you know, they both are saying, okay, yep, we're friends, we're not foes. We, you know, okay, I guess we're good to go here. And so there has to be a way for the bacteria to sense where these plants are. And of course, they can't talk to each other. So it's going to be chemical communication that seems to be happening where the roots are excreting out these chemicals as it's living its life and just like, you know, kind of throwing it out there like, hey, you know, any nitrogen fixers here? And so these chemicals get secreted and the bacteria has to pick up on that chemical gradient and be able to follow that to the root surface. Depending on the plant, because of this very tight specificity, there's different chemicals depending on which plant you're looking at that will be sent out to alert the bacteria, hey, you know, I'm here, come on over. And so this positive chemotaxis is the idea that the bacteria are sensing that chemical signal and they work up a concentration gradient of that signal in order to get to the root surface. So positive chemotaxis by the bacteria. And then in pea plants have probably been maybe the most well studied in terms of this dynamic. And they know this chemical, homoserine, it's a homoserine, it's an amino acid type structure. And that's what the plant's secreting. And that's what the certain species of bacteria picks up on. So that can be one idea.
So if you were to if a different plant were to produce that chemical, would that bacteria then be able to produce that relationship?
Yes. So if there's two different plant species, but they're both producing, for example, homoserine, yeah, the bacteria would be able to, that's the signal the bacteria pays attention to. It's got the receptors for that, you know, on the cell membrane of the bacteria. And so when it docks into the receptor, that's like the signal, you know, the telephone call to the bacteria. Oh, okay, here's a homoserine. That's the one I'm adapted to. So I'm going to move toward the root, follow that signal to the root surface. Flavonoids are pigments, and flavonoids also are excreted by roots of certain species. And we'll learn more about flavonoids as we get into photosynthesis because they, you know, some of the brightly colored parts of the plant, the flavonoids actually are playing a protective role because of too much sunlight. But anyways, here in this case, it's actually a signal molecule in the soil that is, you know, attracting these nitrogen fixers to come to the root. Okay, well, that's only half the conversation. The plant is pretty continually sending these signals out, you know, alerting any microbes in the area to come on over. But then once the bacteria gets to the root, the bacteria has to send a signal back saying, it's okay, you know, you called me, I'm a friend here, let's hook up and form this relationship. And so we call all the different signals that different bacteria produce for this, we call them nodulation factors. And so it's just a kind of a general umbrella term. And there's different nodulation factors that, you know, sometimes we just shorten it down to nod because it gets tiresome to say nodulation all the time. So you'll hear people talk about nod factors, all right? That's what they're talking about. So the bacteria will send back a nod factor to the root once it gets to the root. And one of the first things that they see happening when they follow the time course is that the host root, its morphology starts to change. So kind of like what we talked about with the ectomycorrhizal fungi, that the presence of the ecto on the root can actually cause the roots to form that bifurcating Y shape. Well, with the nod factors, it causes the root hairs to start to curl. So the tip of the root hair, instead of growing out straight, it starts to curl, and I'll show you a picture of that. And then some plant species, the root hairs will actually start to branch and not just curl. And so that's one of the first things they notice when they're doing time studies of this in the lab. And I like to think of especially the curling, when I see images of this, I often think that the plant is almost like trying to hug the bacteria in close so that they make good physical contact, so that the bacteria can do the next step. And so that's just a way for me to remember the process. It's like they're hugging. So, and here that's what I mean. So, and they actually show this under, really nice imagery, stained imagery, that the root hair curls like that, and that's where the bacteria kind of, there's more bacteria in the little crook of the arm there, so to speak. So that's what makes me think of them hugging. So here you have the rhizobia bacteria, just getting to the root and secreting a nod factor saying, hey, I'm a friend, let's get going. And then that causes the root hair to curl, kind of hug up the bacteria. And then the bacteria will do their next step after that event. So here they're just showing the root hair and a cluster of bacteria on the surface when they first meet up, right? So certainly chemical communication has to happen to get them to meet physically and make contact. Here are some nod factors. And again, they have this central portion, they bracketed, and then they put N down there. That just means that this can repeat. Depends on the nod factor, the bacteria doing it. You can have a long chain or shorter chain. So these are some examples. So nod factors are a group of a chemical class called the lipochitin oligosaccharides. So oligosaccharides just means, a short chain of sugars. Lipochitin, so lipids and chitin involved in that overall structure. It's not you don't need to know that to any detail. Nod factors, just know nod factors are produced by the bacteria. Okay. So now let's imagine that root hair has curled. It's kind of captured some of the bacteria into the crook of the curling there. And so now what's going to happen? Well, as the bacteria are secreting those nod factors, that will get docked into receptors that the plant have on the root. And they're called lectins. So lectins are just protein receptors in this case that the plant has. And these protein receptors, the nod factor docks in to the active site, and that is like the signal. That's the way that the nod factor is conveying information to the plant. So once that nod factor docks into the lectin protein, then that's the way the plant goes, oh, okay, you are one of my friends. I want to connect with you and get established into this symbiosis so we can fix our nitrogen. So they're just receptor proteins that are produced by the plant. And it really is, it's a very specific, you have to get the right docking going in. If it's the wrong chemical, it won't dock in correctly to the lectin, and you won't get the conversation to be finished. And so that's really what they point to when they say this is a very specific interaction. It's, this plant species and this species of bacteria that's going to be able to do this successfully, all right? Okay, so then when that happens, that's usually when you see the rhizobia making really good contact with the root hair and that curling action going on by the root hair. And then they'll get into the next phase. Your book has this figure in it. I'm not going to get into the details of this. I put it in here though to get you to realize that, well, first of all, you can see the lectin protein here and the nod factor docking in. So it's a nice visual to get you thinking about the receptor lectins. And then I just want to let you know it's more complicated than what I'm describing. You see this arrow into the nucleus of the plant root, and you see that there's these calcium fluctuations in the nucleus. So they're looking at this really intensely because they want to understand this down to the nitty-gritty detail, because they are very interested in creating more of these symbiotic relationships with other plants that don't have the ability. They're really looking into this. They want to make plants, genetically engineered plants, to be able to do this with, pair up with key microbes that are doing nitrogen fixation. So it's more complicated, obviously, than what I'm getting into, but this also has a nice visual of the idea of the nod factor docking in, okay? So the next step will be invasion. They call it invasion. I think that's a poor choice of words because it really is a mutualistic association, but the bacteria have to invade into the roots. So we'll do that next, but we've run out of time. So we'll talk about that on Wednesday. So remember, Wednesday's lecture, I'll start with talking about the light reaction of what you're going to do in lab, and then we'll get back into nitrogen fixation.
So Lias one, yellow stripe one, and maize.
Really?
Yep. for iron
And it's really called that?
Yep.
Oh my gosh.
It's in corn.